Good afternoon, guys. It's Saturday afternoon. I'm going to try to go over chapter 15 for y'all. Hang with me. Let me pack it up. I actually was looking back here. So I hope everybody's having a good weekend. I'm going to go, I know we're going to try to get these copied, do the video of the lectures, but I was not able to get with my gentleman that does this in Northwest. And so I'm going to go ahead and do a little, I'm going to try to do 15 as quick as I can to give y'all just some thoughts on this. If I'm going to follow the train of the book, but if I see something that may be tricky, I'll slow down and talk about that a little bit. Okay. So we make sure we're fired here. Make sure we're recording. I'm going to turn on my screen now. And you should now see the PowerPoints for special senses chapter 15. We had all four of our classes this week kind of slightly hit over them a little bit. We kind of quickly looked over the main groupings here, the special senses. If you remember, we talked about this, how there's general senses, touch, pain, temperature, et cetera, et cetera. Special senses, though, are taste, smell, light, sound, and then this static and dynamic equilibrium where we're talking about just a little while, the body motion and head movement, the sensation of movement, and our body orientation, okay? It's kind of interesting to know that smell is the only one that goes directly to the brain after it leaves the olfactory neurons, instead of having a cell or so to hit in there. The special senses, that's kind of unique. Okay, very quickly we looked at the smell here. This picture is pretty cool. In the top of your nose, you have the olfactory tracts, cranial nerve number 1 sitting on the cribriform plate, the ethmoid bone on each side of the cristogala. These little holes here allow the nerve to come through, and there's an epithelium in here that contains the receptors here for smell to occur. Okay, now check this out. Now, you have to have atmosphere for to have smell because the air has to rise, and then once the chemicals in the air get up in the nostrils, they mix with moisture. Smell and taste will not work unless you have the chemical in solution. So you have to have water. A completely dry tongue, completely dry nose, will not smell anything or will not taste anything. Okay, kind of wild. I would say that as we look at all this, set us in class this week, think of all the animals in these 5 special senses. We don't have the best of any of these 5, but we have a good mix. Obviously, the dogs, and the bloodhound would have 1,000 times more powerful nose, and they can actually smell chemical trails that you leave as you walk out of a room. Okay, pretty wild. Okay, not going to get into too much of the physiology here. I want to get on to the hearing and vision. We like to cover this semester, but you can see the chemical in solution binds receptor, triggers a second messenger, acetylcholine to be produced, cyclic AMP to be reduced. That triggers a sodium channel to pop open, thereby firing an act potential that would then go to the brain as a smell. Okay, not a whole lot on that one. Here's a condition where you cannot smell. Of course, we got a lot of people with COVID-19 right now. They're losing smell and taste, and that's pretty wild, right? And some people have reduced smelling. I think I probably have this one here since I have dissected cats for a living for 30 years in AMP labs. And was raised on a dairy farm. I have a very low tolerance, don't smell very well, things very well, and nothing kind of grosses me out. Okay, on smell. Okay, moving on. As you get older, you do experience more of this, and once you get in your 60s, you will have gradual loss of smell. Okay, taste very similar on these papillae and the tongue. Take a look. So taste receptors are around the edges in the back here, not a lot right in the middle, but right back in here and along the sides. And the chemical in solution goes down, binds to receptor, triggers an acts potential, and then Shazam, we get the taste. Okay. Worth noting here, I was going to go back and read this. I'm talking about smell here. Check this out. Humans can detect about 40,000 different odors. We only have about 350 genes coding for receptors. Animals can just do way much better. I'm just looking. I thought I had a note in there somewhere. Okay. We're typically born with approximately 10,000 taste buds, but lose half of them by the age of 50. So my taste is going bad. Okay. Anyway, on that. Now, the fun part about taste is that we have 5 distinctive receptors. We have sweet taste caused by simple sugars. Sour taste is produced by hydrogen ions, for example, found in lemon juice. Salt is sodium, potassium, metallic ions. Bitter is nitrogen-containing compounds. They're very bitter. And then umami, which is the latest one discovered by I believe the Chinese, Japanese, that is translate that word means simple, delicious, or associated with broth or meat, sushi kind of taste, and it's a glutamate amino acid that triggers that. Okay, I still don't get metallic. I think it would have to be another one we haven't discovered yet, but we'll see. Or I just can't quite get sweet, sour, bitter, and salty and make metallic. But that's just me. Okay. Fun one here we talked about in class about super tasters. They're those who taste things very well. We used to do this little chemical in class. A better one would be if you could, if Diet Coke tastes weird to you, doesn't taste like regular Coke, then you're probably a super taster. To me, it tastes the same because I'm in this group right here of the mild taster. I'm on this chemical. I'm in this group. I don't taste anything bad. It just tastes like a piece of paper. But you can buy this little piece of paper, have this on it, and the fourth of the class tastes nothing. Half the class feels something like a sweet bitter tart, like a piece of candy. 25% won't throw up. So it's kind of fun to do that in lab. Okay. Moving on how it fires, you can see the chemicals bind. We have an axe potential. Shazam goes down, fires, and next neurotransmitter released in the synapses. So nothing. I'm not going to get into the physiology of all that. Here's how it goes into the brain. Don't know how I would test on it. But you can see the parts of the brain it goes to. Okay, let's get on. I want to get on into the eye, which is super cool. Okay. Going on through all this, the eyelids, et cetera, et cetera. You can see all this. We have, I think one of the cooler things about it is that we have the conjunctive, the eyelid itself, right? It's a conjunctivitis would be an infection of the eyelid. We have a tear duct apparatus, this lacrimal apparatus. The lacrimal glands are always making tears, and we're collecting them, okay? And they lubricate, wash away debris. They just nurture the eye. But look at this x-ray. You have a drain here on the inside of your eye that goes into your nose on both sides. There's an x-ray with dye injected in it to show it. And you can actually, if you learn how to do it, I've seen people that could blow milk out their eyes, okay, through that. But of course, I have had some friends that have had to have surgery because this got clogged. And I had a friend that had a dog that had to have this fixed on because they were crying all the time and very expensive for dog surgery, okay? We got the muscles here that move the eye. Not going to really go into those. I won't get on into parts of the eye. I'm just kind of doing a quick version of this. But if you look to the cross section of the eye, you have the cornea, which is a clear outer purpose of the eye where you put a contact lens on. Then you have the front chamber called anterior chamber full of aqueous humor. Then you have the pupil, which is the colored parts of the eye, but also controls the diameter of the pupil. So when it constricts, it closes the pupil down, letting less light in. When it contracts, like at night, it opens up and lets more light into the eye. Then we have the lens. It's going to focus the image onto this thin yellow layer called the retina. Under it is a choroid coat that is behind it. That's usually black in ours that absorbs any light that goes through. In the deer and animals like cows and stuff, it is a shiny layer called oh man, it's slipping my mind today, but we'll talk about it when we cut those eyes. But in the lab, but it is actually reflective, and that's why they see better at night. That's why animals have better night vision than we do. Their eyes, the back of their eyes reflect light back up onto the rods and cones, giving them a little bit better night vision. The whole back port is held in place by a jello type material called viscerous humor, okay, and that holds the retina down. And then you can see here that the optic nerve is where the retina comes out. There is no retina here where the blood vessels come in and out and the nerve leaves, and this leads to the blind spot right here. So each eye, in each peripheral vision, you have a blind spot just a little bit away from where we have the center of focus called fovea centralis. So when you're looking at somebody, your eyes are moving so the light hits the fovea centralis coming in, and that's where you have your center of focus, and that's where you have the most cones to color and fine detail in the eye. We'll talk about that later, but the retina has two photoreceptor rods and cones, and the fovea centralis is the center of focus, so it has the greatest concentrations of cones, okay, which do our vision, okay. Goes over the sclera, the outside white part of the eye. We done talked about the cornea, where you have astigmatism, uneven curvature of the cornea or lens, where you would have to have glasses or have corrective surgery. The lens is held in place by suspensory ligaments attached to a muscle called a ciliary body, which is not labeled right here, but it's this muscle right here is a ciliary body. And this can relax and contract and change the tension on the lens that allows us to accommodate and thereby focus on objects, to allows the eye to focus on objects close versus far off by releasing and contracting the suspensory ligaments by relaxing and contracting the ciliary muscle. Okay, there it is. The ciliary body is that muscle mentioned there. Sensory ligaments, got that. Iris, color part of the eye, controls amount of light. Pigment is the hole itself. Here's another blow up. There's the ciliary body, ciliary muscle there. You can see it, how that all is put together. Pupillary sphincter muscle, right? That is going to have, parasympathetic nerves is going to cause it to close down. And when you get excited, they dilate, right? We talked about that last semester. So decreasing light, it opens up. But when you get scared, it pops open. Okay, that's pretty cool. 10 layers of the retina with the photoreceptors, rods and cones. Cones do color and high detail, but to have bright light. Rods do black and white and low light vision. This is why at night everything's in shades of black and white, because rods do night vision. Okay. Fovea sensata, already mentioned that. Optic disc is also the blind spot because there is no retina where the nerve and blood vessels leave the eye. Here is a picture. You may have this done. I had this done last month at my optometrist, and they're looking at the blood flow here. This is a good one here, but I actually have a little folding here in my retina where it's pulling off a little bit, and they're keeping an eye on it here. So here's fovea centralis, and there's the blind spot, optic disc there, and that red is all the arteries and veins carrying blood to the retina. Here's a cool one here. We'll do this in the class. Demonstration of a blind spot. You can focus. I'm not going to work through, but you can focus on this and make the dot, make the pencil fill in or the dot disappear. So there is a blind spot. Okay, and we'll talk more about that. Same thing here. Kind of gone over most of this. Nothing much new here. Cataracts is a clouding of the lens, right? And it can be replaced by having an artificial glass lens put in. Very common in older people. Okay, glaucoma is another one where we have a pressure buildup, and it's coming in a minute here. Talking about the cavity flow, where that posterior chamber has the viscerous humor, and the anterior chamber, the anterior cavity has that aqueous humor. Okay, and that flow comes through here, so we make the fluid and it flows through. And if it doesn't properly drain, we can get glaucoma. Where the aqueous humor doesn't drain properly, fluid builds up in the anterior and posterior chamber, and it squeezes down that jello onto the retina and kills the retina. Compression and damage to the retina and the optic nerve. Second leading cause of blindness can result from eye infections, certain medications, congenital defects, or in most cases causes is unknown. So you don't know anything's wrong, you go blind. So you do need to go to the eye doctor and have that pressure checked. And that's when they put the little pressure gauge on your eyes to tell. They just gradually lose their vision. And once they go to the eye doctor, there's nothing that can be done. We cannot replace vision, cannot be restored on an eye that has a glaucoma. So pretty wild. Now, I think I said this earlier, but well, an extra idea, but when we get down to things that we don't understand completely in the body, I mean, we've got a lot of the muscles and bones figured out, no new bone discovery, but no one's explaining to me how the brain takes vision and we get this perception of what y'all watching here. So still some mysteries left here, but the brain's ability to do this perception of vision is still one of the great ones that I don't understand. Okay, but we do know this. Okay, that light is emitted from the photons of light, of energy. Basic unit of light is photons emitted from the sun. This is what a photon looks like. So it contains all these waves, gamma rays, X-rays. As we go right, the waves get bigger and less dangerous. So the gamma rays are short wavelengths and very dangerous. X-rays, ultraviolet. And then you have between 350 nanometers and 750 nanometers, the visible light spectrum. So if you put your clothes in a color ordered in your closet, then you need to reorder them in this order. This is the order of nature of light. Purple, blue, green, yellow, orange, red. That is the wavelengths. And so we have different photoreceptors that detect the different wavelengths of light, thereby giving us the different colors in our vision and our mind. We can't see past 750, infrared, spectrum, microwaves, radio waves, et cetera, are farther out to the right. Okay? So light then is also reflected, okay? And so air has a reflection index of 1. So light passing through air is not significantly bent. Water has a higher refraction index, and you've all seen this because when you reach for something in water, it's reflected, it moves. That's due to the density being greater than air. So we got a problem with vision. We're gonna have to be able to correct some of this, okay? And so one of the things we can do is reflection of light also depends on the angle that light strikes surfaces of objects. The greater the angle causes greater refraction. Curved surfaces bend light rays more at their edges. So convex lenses, surface that bulge outward in the middle, cause light rays to converge or bend inward. And then a concave lens will spread the light out, okay? So clear vision requires that light rays are focused on the retina, not in front or behind it, okay? So 2/3 of eye reflection power occurs as light passes the cornea, okay? And then the lens provides the fine tuning. This is where it gets weird. Okay, normal eye is relaxed and focused on a distant object. This is called this atrophic state. Okay, light rays from objects closer to the eye are more scattered, need more reflection. And this occurs when we try to focus on something close, or what's called accommodation. So looking at this apple from way off in a tree, then everything's fine. The rays are going right on here, and it's beautiful. But if you get the apple up close, then we must round the lens off, has to round up to bring the convergence in, so that we can reflect the right in, so you get the right focus. So accommodation is when the lens gets more round for near objects. And to do that, we have to have the slack taken off of the ciliary muscle, and the suspensory ligaments come loose, and that causes it to swell back out. Okay? So this throws a lot of people off. So ciliary body surrounding the lens relaxes when viewing distance objects. And that causes the pulls, sphincters like spoon butt muscles away from the lens, creates tension on the suspensory ligaments, and they flatten the lens, like so, like this. So note when ciliary muscles is relaxed, it opens. It is open. The sphincter is open, places farther away from the lens distance, and from the lens puts tension on the suspensory ligaments, and it flattens the lens. So that is when we're looking at things far off. Okay. Then now, when we look at something close, the muscle relaxes, takes the tension off, but that causes the lens to swell out. And it throws a lot of people off. So it rounds off so you can see things up close. Okay. Matter of fact, they talk about that right here. I'm going to back it up a little bit. So talking about two events happen: pupillary constriction and convergence. Okay. Movement of ciliary muscles seems counterintuitive because suspensory ligaments tighten when muscles relax. Generally, we associate this kind of tension with muscle contraction. But recall that sphincters or circular muscles usually control the flow of material in and out of an opening. So they open, so when they're relaxed, they open, but when they contract, they close. So maybe that'll help you see it. So she does a good job here. So when they look at it far off, the muscles are going to what? Take a look. Take a look here. So notice when ciliary body muscles in a relaxed state, that creates tension on this, and that flattens the lens out. And when we look at something close, the muscles relax, putting slack on these, and allows the lens to bubble back out to focus it on. Now, we're going to do this in a lab, possibly called a near point of accommodation. And this is just how close can you hold the book to your eye and still read the text? Well, it's usually around 10 to 12 centimeters for people under the age of, so you get around 50. And after 50, it gets farther out. So if someone's 70 or 80, it might be 100 centimeters. So when I got around 45, I had to get reading glasses or bifocals, and now I have to have them just to see anything. Okay, so we'll do that in lab, and that's called near point of accommodation. So this actually shows you how glasses work. So if you have farsightedness, hypertrophy, farsightedness, you're actually focusing out of your eye. You get a convex lens, and it pulls the light rays in to focus on the focus centralis. Conversely, if you have a squatty eye and your eye is not long enough, your focal point may be here, then you would get the concave lens to push it out, the focal point out farther. So lenses, that's for nearsightedness. So lenses have done great. I think one of the great miracles of all times is glasses, right? By adjusting that, we can fix the shape of your eye by adjusting the lenses in your glasses. Astigmatism, I already said that. Uneven curvature of the lens of cornea. The astigmatism of the cornea can be fixed with this laser-assisted in situ keratinosis, LASIK surgery, where they burn off little grooves and it lets it smooth out the surface of the cornea. Okay, and that's pretty wild. Okay, and then we get into our photoreceptors. Remember, cones, bright light, high resolution, color vision, rods, low light but motion, and what they do that to detect color. Okay, here is a drawing of a retina, and here is the actual 10 layers of the retina. You can see them labeled there. The rods and cones, super important in here, right? Rods are rod shaped, cones are cone shaped. Okay, the rods way outnumber the cones. I was looking that number up because my memory is not working super good today, but pretty fascinating. I'm going to pull it out here, right here, real quick. But like I said, it is way outnumbered here. Let's see, where's that number? You have 100 million rods and 3 million cones. So 3 million cones in an eye, 100 million rods. So cones are way outnumbered by the rods, okay? Now, there's a picture showing rods and cones, electron microscope of it. There are blue cones, red cones, and green cones. So there are three main primary colors there. It can give us all of our colors, okay? I'm not going to go, I'm going to go skip over the summary on this because I just want to get on there. I'm not going to go crazy on tests. But look at this. In the dark, the retina ganglion cells are not stimulated in the rods, okay? So notice this. So the photoreceptor depolarized, releases glutamate onto the bipolar cells. Glutamate inhibits the bipolar cells from releasing neurotransmitters. No image is fired. Light hits a rod. The light hyperpolarizes the photoreceptor, stops releasing glutamate. The bipolar cells are freed from inhibition, and now they fire action potentials that release a neurotransmitter that fire to the brain as a particular color or wavelength of black and white, okay, or as a wavelength of light. So this is how vision works. And like I said, how that all gets together in the brain is beyond me. Color blindness, 8 to 10% of male are colorblind for one of the colors. Women, less than that, less than 1%, because it's on the X chromosome, and boys only have one. So therefore, it's an X-linked recessive disorder. Females have 2 X's, so you can have one bad gene, and the other X give you normal vision. But boys only have one X, and if we get one that's missing a color receptor, then we are colorblind for that color. That's pretty wild. Quick check here. If you don't see the, was it 74 there? I guess that, or 94. I can't make it out. I think it's 70, whatever that is. And that 2 numbers there. Okay, 74, I believe. Yeah. But look at here. In the plate below, person with red-green color blindness will not be able to detect the number 74 and will see 21. So I am barely seeing that 74. Okay, so I'm having some issues with that myself. Okay. Moving on here, it just talks about the way that the left eye goes to the right side of the brain, right eye goes to the left. And there's this area on the left and right that each eye, only the right eye sees this, on the left eye sees that. Okay, that's kind of cool. Nothing much more we can do about that. Moving on, moving on. All right, and I think, let's see here, we may go ahead and try to run through the ear real quick. This is going to turn into a long video. Parts of the ear, the pinna, the outer part of the ear canal, okay, and then the tympanic membrane. Then you have the 3 smallest bones in the body, the malleus, the malleus incus stapes, 3 little bones in the body. We have them in the lab on a little glass model or in a piece of plastic. You can look at them. And then you have the semicircular canals here that looks like a seashell, where hearing is going to occur, in the organ cortae and the cochlear. Okay, and then you have the vestibula that is going to have structures that allow us to know basic body orientation without motion. It's called static equilibrium. And then the semicircular canals here have fluid in them that are going to slush, that are going to allow us to do what's called dynamic equilibrium and detect motion of driving, or the sensation of driving down the road. I always say riding with my daughters when I was trying to teach them how to drive, and they'd hit the brakes and all that, or accelerate. And that is achieved with each ear's semicircular canals. Okay, we're going to real quickly just take a shot at hearing, which I think is one of the coolest things ever. And we do understand how that works. Hearing works like a piano with all those different keys, okay? And the piano keys are in this organ of cortae and the cochlear. So sound waves are going to come in, and they're going to cause the tympanic membrane to vibrate. And then that hits the 3 bones, and they pop each other, and they hit. And that middle one, the last one, the stapes, pops this oval window in the semicircular, in the cochlear. This causes waves to occur. So we convert sound waves into hitting the side of a fluid-filled drum and causing waves in this drum. And what makes this drum really cool is that inside this structure, there are 3 chambers, and all 3 chambers coil all the way to the inside. Now, if you uncoil this thing, the 2 chambers come together. The outer and the inner chamber will come together. So, and I'm going to show you that in just a minute. So I'm getting ahead of myself. It's very cool. Okay. So we got the earwax, blah, blah, blah. Talk about that later in class. Okay. Audio tubes, eustachian tube, pharyngeal, tympanic tube, that keeps the pressure between the throat and the ear. So when your ears pop when you fly in an airplane or go down a hill or mountain, that's snot going between the ear and the throat. Okay, pretty wild. The three bones, there we go. More on that. Let's keep on. Okay. Inflammation in the middle ear can cause problems in kids, and they put the tubes in the ear to let that pressure out. That's why chewing gum when on a plane, taking off and landing keeps the ears from hurting. Here is a kid that had the inflamed otitis media, and here is the tube to let that out, so they can let the pressure out. But these tubes do eventually fall out. Okay, and we treat that with antibiotics also, right? Okay, let's look at the cochlear or whichever vestibule. Let's get on. Here's what I was talking about, the fluid flow. Semicircular canals have fluid in them, and each ear has a tube that does this angle, and then cuts the angle. So any movement you could possibly make with your body is going to make fluid in each ear slush a different way, and that slushing hits a receptor here at the crista ampulas right in here, and that causes a pattern of action potentials to your brain that tells you that you're moving left, spinning left, right, falling, rising, etc. Okay, and we'll see in the uricles and saccules of the vestibula, we're going to have structures that do that static equilibrium. But notice there's that chamber I was telling you about. That there's a chamber all the way through that cochlear where hearing is going to occur. And of course, there's the optic nerves, the vestibular cochlear nerve, not optic, cochlear vestibular nerves coming out, going to the brain to do hearing and equilibrium. Okay, now they don't do a real good job here. Let me show you. The chambers are the cochlear duct, scala vestibular, and the scala tympana. There's a cochlear duct in this ear. Now, here's the deal. If you take this thing and slice it open, remember it would unroll like a fruit roll-up, kind of like a biscuit thing, like a sheet of dough. But if you look, if you roll it all the way, let me go there first. If you roll this thing all the way out, take a look right here. There is that whole thing rolled out. So if you unroll the cochlear, you would have the scala tympana on the bottom, scala vestibular where that's getting hit by the stapes, and that wave works all the way to the innermost of that, and it can work itself out coming out this round window. But the organ of corte is in here. So as the waves hit here, we get a depolarization or pushing of that membrane up, and that pushing of that membrane up is going to allow receptors from the outside, from the outer part of the coil. High frequency is here, and the very middle of the coil is low notes like this. And they're in this organ of corte here, there are hair receptors that get touched that go to the brain and tell us when a 20 megahertz deep bass hits or a high frequency airplane taking off. Dog whistle is like 22,000, which we can only get up to 20. Now, look at that. Now, I'm going to take a slice of that. This is what a slice of that looks like under a microscope right there. So that's one of those little pieces. There's the scala vestibula, cochlear duct, scala tympana. And in that right here, there's that tectorial membrane. There's those hair cells I was telling you about. So basically what you got there then is a keyboard going from 20,000 to 20. And when you cut it, it looks like that. You get a whole bunch of these, right, which is very confusing. You get all these individual chambers, right, because that is that single layer rolled up to the middle core where the scala vestibula and the scala tympana fuse together. Okay, pretty wild if you think about it. Okay, and I'll try to show that better in class, but that's a hard concept to see. But if you unroll it, it makes more sense. And different receptors detect different frequencies of sound. Okay. All right, so what is sound? Okay, well, sound waves are being produced, right? Okay, the trick with that sound must be understood and truly appreciate hearing. Ears detect and transduce the sound waves into electrical signals to the brain, okay? So, two things you need to know. Every sound has a certain pitch. That determines how many times objects vibrate back and forth during a certain period. It's measured in hertz. Human ears can typically detect sounds between 20 and 20,000 hertz. That's the cycles or vibrations per second. Okay, loudness or amplitude is how loud it is, and that is measured in decibels. Greater force hitting the drum makes a greater difference between the waves of high and low air pressure producing louder sounds. And you also must have an atmosphere for sound. I hate to break it to everybody that loves Star Wars and all the sci-fi movies, but all that and blowing up is dead silence. There is no sound in space, okay, because we don't have an atmosphere. Okay, so here we hear plucking the string here, and you get the vibrations, and those vibrations, the wavelength, therefore the pitch, and how high from high to low is its loudness. Okay, so once again, you can read through this. The sound waves then are converted to vibrations. Those waves cause that tectonic membrane to touch up and to fire these hair cells right here. But the point I want you to get is the hair cells are laid out in that coil from 20,000 to 20,000 to 20 megahertz. Now coil that up. So bases are way back in here, and as we coil our way out, a vibration causes the tectonic membrane to touch that hair cell at each point. So 20,000, all the way down to 20 in there. Now, when I first started teaching physiology labs at Ole Miss years ago, we actually had an experiment we did where we put on little headphones with a computer, and we would play 20 hertz. It'd be like, then we go to 1,000, go to 20,000. I mean, we'd go to 20,000, I didn't hear it. And I thought the other guys were messing with me, the other lab TAs. So I grabbed the controls and I turned it up really loud. And I'm going to do this in lab, in lecture. And I played 20,000 megahertz, or about 18,000. I can't hear anything above 18,000. So I got a recorder, tape recorder, and copied that, and made that sound so I could go around and ask people what it was. Took me a little while, but that is the sound of 1155 massive first tractor at full throttle. So as a child from like 13 or 14 till 30, I drove tractors, and we would go full. We didn't have no cabs, we didn't have no headphones, and we just listened to that loud tractor. And then we had a special, really cool tractor that had radios on the right side, so my right ear is worse than the left because it would blow music louder than the tractor to the right ear. And so I destroyed my hair cells at 20,000 megahertz. And I'll show you in class a picture. I don't have time to Google it here. But so one of the worst things you can do to yourself is expose your ears to loud noises because you will destroy the hair cells and we can't fix it. And so when I lay down at night, I hear ringing in my ears. This sounds like the static on the TV when the station goes off. Popping sound. Okay, and that's my fate because I've destroyed that. I beat those hair cells to death and they're just firing randomly now. Okay, pretty wild. So there's a hair cell where it touches, and maybe it's an electron microscope picture. Here's what they look like a little bit better. So when they touch, they fire an exponential as one of those notes from 20 to 20,000 megahertz. Okay, and there it is again. There, neurotransmitter release goes to the brain as a note. Trying to see if we got anything else. They're showing. I'm not going to go into how it processes the brain. She talks through that. Here's what I have: tinnitus, whatever pronounce it. Excuse me. That is damaged stereocycle, damaged hair cells, cochlear hair cells, and I have that ringing in my ear. She should have put a picture in here of that. I don't think we did. I'll show you one. People who have no ear nerve connection can have a cochlear implant, and we'll talk about that in class. But I have a friend of mine who has this, and they were able to hear. And the big thing about this would be, and there's a big debate among the people born deaf, but they don't like these implants. A lot of them don't. A lot of the deaf people don't like people getting implants because they said, I'm born this way and I'm going to live with it. But to me, if you can get a chance to hear, you should, because you can't have speech without hearing. So now you say, well, I know a friend that's deaf that speaks some. No, they got deaf as they got older. But you must have hearing to learn language as a child. Okay, then they learn other languages, but they can do, obviously Braille and other things. But that's the truth, and I'll talk more about that in class. So big debate if it ever happens to your kids. Okay. So that kind of throws it all together how that works. Okay, let's do these other ones real quick. So we got the other two sensations of the ear, and here we're talking about the vestibular and the semicircular canals, and static equilibrium and dynamic. So the tilt of the head, maintaining balance, is static equilibrium, and then the semicircular canals, that fluid slushing in those tubes, do rotational or angular motion called dynamic equilibrium. This is one of your short answer questions. Tell the difference between static and dynamic equilibrium. Okay, the uricles and saccules here. Now if I made this up, you would think I was lying, but look, in this structure right here, you have a layer of hair cells with a layer of gelatin down. And then on top of these are little rocks called othellets. And as you roll your head forward, the rocks roll to one end, and they cause the hair cells to fire neuron activity that the brain detects as the head's tilted a certain way. Is that not crazy? You go into outer space, these things float. That sends a pattern to the brain that you don't know. The brain don't know what to do with. And so when the brain's in doubt, let's just throw up. And that's motion sickness. That's where about half the people going out of space, 75, half to 25% to half throw up when they go in because they get a misreading to the brain that they can understand. Same as motion sickness over here. These tubes are full of fluid, and as they move, they move a neurotransmitter head in here in the crista ampulla, and that all detects motion. So if you're in the back seat reading the paper and you're going up and down hills, and your semicircular canals tell you you're moving up and down, but your eyes say you're not, then the brain gets misinformation, or it can't make that work out in its brain, because if one says, hey, I'm not moving, eyes say I'm not moving, semicircular canal says, dude, let's just throw up to be on the safe side. And that's motion sickness. Okay, so look, as you tilt one way, the rocks rolling one way, and the stericycle fire and tell the brain the way you're going. And then dynamic equilibrium, in these crista ampullas, the fluid blows by and will move these back and forth, left and right, that then tells you which way you're moving. So you get this feeling of riding a roller coaster. Each ear being separate apart a little bit will have a difference between firing, and that tells you which way you're moving or spinning. Pretty fascinating, yeah. So rotating the head will cause the fluid to move a certain way that the brain detects as I'm turning my head left and right. Motion sickness is mismatched sensory information from the eye and the vestibular structure. I'll tell you in class, a friend of mine I met a couple years ago to AMP convention took an antibiotic and killed all the hair cells in his vestibular and saccule, so he has no static equilibrium. He always touches the table, holds on to things, so his body knows which way's up and which way's down. Pretty fascinating. They have some patches they can give. Some people get real bad, vertigo, which is a firing of these parts when you're not moving, and that's a big problem too. Okay, I think that's about all I got on that one here. Nothing much. I'm not going to do much on speech, but I think those are the parts of the brain where smell, taste, vision, hearing, and vestibular sensation go. That's pretty cool. All right, guys, that's a quick overview. We'll hit some more of it in that class, and I'm going to definitely have a test question on how do we hear, and I'll go over that a little bit better in class. Y'all have a good one, guys. Bye.